Good afternoon. Welcome to Pavitra classes. Today we are studying about another important topic that is mutation. Mutation is very important in very biotechnology subject. PCI syllabus biotechnology. Unit 3 mutation. Mutation means sudden heritable change in the genetic material. As we know, gene is the characteristic material, genetical characteristic material of our body. Sudden heritable change in the genetic material or character of an organism is known as the mutation. So sudden heritable change may be from physical, chemical or environmental changes, but the permanent sudden change in the genetic material is called as the mutation. Individuals showing these changes are known as mutants. So the genes which got altered are called as the mutants, and individuals showing the altered phenotype due to mutation are known as variants. So which is change in the previous gene material, genetic material, so that change alteration in the phenotypic material, that is called as the variant. Factors or agents causing mutation are known as mutagens. Mutation which causes changes in the base sequence and gene are known as the gene mutation or the point mutation. These are the some of the definitions. So what is mutation? Sudden heritable change in the genetic material or character of an organism is known as mutation. Individuals showing these changes are called as mutants, and individuals showing an altered phenotype due to mutation are called as the variants. Factors or agents causing mutation are called as mutagens, and mutation which causes changes in the base sequence of a gene are known as the gene mutation or a point mutation. So coming to the history of the mutation, English farmer Seth Wright recorded case mutation first time in 1791 in male lamb, usual short legs. So male lamb animal, he seen the animal with the short leg animals compared to others, that causes the due to the genetic material change, alteration in the genetic material change. In 1791 when he is doing the research in animals, he checked this. The term mutation is coined by Hugo de Reis in 19,000 by his observation in Ornithera. Systemic study of mutation was started in 1910 when Morgan genetically analyzed white eye mutant of Drosophila. Next, later H.J. Muller induced mutation in Drosophila by using X-rays. 1927 by using the rays he checked again mutations. So he was awarded in the Nobel Prize in 1946. Coming to the characteristic of the mutation, generally mutants alleles are recessive to their wild type and normal alleles. Most mutations have the harmful effects. Because of that, some mutations are beneficial. Spontaneous mutations occurs at very low rate. So very spontaneously changes will be environmental changes or spontaneous changes at very low rate they will occur. If we want any mutation or altered genes, we have to give the external applications. Some genes show high rate of the mutation. Such genes are called as the mutable genes, so which are very eagerly get the mutation reaction, that is that genes are called as the mutable genes. Highly mutable sites within a gene are known as hotspots. So where the area in that gene where which genetic code is very fastly getting mutated, that called as the hotspot. Mutation can occur in any tissue, any cells or any of the organism. These are the characteristics of the mutation. Next, coming to the classification of the mutation. So many classifications are there. So how classification is based on which type, that is very important. First classification is based on the survival of an individual. So first in that 5 classification are there: lethal mutation, sublethal mutation, subvital mutation, vital mutation, supervital mutation. Lethal mutation, when mutation causes death of all individuals undergoing mutation are known as lethal. Lethal means dead. So after the alteration or of the genetic code or genetic material after the mutation, if that individual organism causes death, that mutation reaction causes the death of the individual, that is called as the lethal mutation. Sublethal means 90% of the death, causes 90% of the death of the individual are called as the sublethal mutation. Subvital mutation means such mutations kills less than 90% of the individual. So less than 90%. Next is the vital mutation. When mutation don't affect the survival of any individual are called as the vital. So it is in the birth only, or it has a organism has a again growth in the cells. So supervital mutation means this kind of the mutation enhances the survival of the individual. So supervital means it gives the power to that cell or organisms with the mutation, that is the supervital capacity. So these are the 5 classification based on the survival of the individuals: lethal, sublethal, vital, subvital and supervital. Next is the based on the causes of the mutation. In that, spontaneous mutation is there, induced mutation is there. Spontaneous mutation occurs naturally without any cause. So we don't know how it is affected. The rate of spontaneous mutation is very slow. Methylation, some of the reactions, deamination of the cytosine. Rate of spontaneous mutation is usually seen in both cells, eukaryote and the prokaryotic cells. Maybe UV rays effect, sunlight effect causing the mutation in the bacteria. This is the example. Second one is the induced mutation. So in this induced mutation, we are inducing some of the rays or some chemical agents like that. We are giving external applications. Spontaneous means within the spot it is showing the mutation. Mutations produced due to the treatment with either chemical or physical agent are called as the induced mutation. The agents capable of inducing such mutations are known as the mutagens. So use of the induced mutation of crop improvement program is known as the mutation of mutation breeding. So whenever crop for the agriculture field, crop improving, any mutation is conducted with the physical and the chemical agent, that is called as the mutation breeding. So X-rays using the mutation in the cereal growing, X-ray light is used. These are the some of the examples. So this is the based on the causes of the mutation. First one is the based on the survival of the individuals based on the causes of the mutation. Third one is the based on the tissue of the origin. So somatic tissues, germinal tissues. So somatic mutation mutation occurring in somatic cells are called as somatic mutation. So where it is that mutation is taking place, if it is in the somatic cell or gametic cell, or in asexually reproducing species, somatic mutation transfers from the one progeny to next progeny. So from one offspring to another next offspring, it is transferring in the asexually reproducing species. So one cell to another cell. Germinal mutation, when mutation occur in the gametic cell or the reproductive cell. So in this sexually reproductive species, only germinal mutation will be transmitted in the next generation. So this is the based on the tissue of the origin. Next is the based on the direction of mutation. So forward mutation and the reverse mutation. When mutation occurs from the normal allele, wild type allele to the mutant allele are known as the forward mutation. So one is the mutant allele and one is the normal. So from normal allele to mutant allele are known as the forward. If mutant allele to normal back it is taking, that is the reverse type of the mutation. So this is the based on the direction of the mutation. Types of the traits affected. Next is the classification is based on the trait affected, visible and the biochemical. So visible is which are affected phenotypic characters. You can observe in the normal observation within the eye only. That is the visible mutation. If that mutation is occurred, the characters are can be seen visually, then it is called as the visible mutation. Biochemical means if the biochemicals product which affect the production of the biochemicals which does not show any phenotypic character or any visible character, but biochemically the mutation affects the characters of the individuals, then it is called as the biochemical mutation. Next type is the chromosomal mutation. In this 5 types are exist: deletion, inversion, translocation, non-disjunction and the duplication. So by altering the chromosomes, these types will be altered. So we will study one by one. Deletion means due to the breakage. So you can see in this picture a piece of chromosome is lost. So if this is a chromosome, this piece is lost, this is deleted genetic material. So again it is rejoined. This is the deletion of the mutation or deletion chromosome. Inversion, so it is the rotated chromosome segment break off So it is first it will be break off, then it is inversion will take place, flip around in the same this, and again reattach to the same. This is the inversion. Duplication, same pattern will be, gene sequence will be duplicated in the other, that is called as the duplication in the chromosomes. Third one is the translocation. So before translocation, like this portion is cut and again it is rejoined here, again it is exchanged. So this is the translocation, involves 2 chromosomes. Last one is the non-disjunction, failure of the chromosome to separate during the meiosis. So in cell division, so mitosis and meiosis stages are there. Failure to that separated chromosomes, again it is not reattaching to that, that is the non-disjunction. So it is not again joining to that, that is called as the non-disjunction. So in this, some of the diseases will be there because the gamete will be having too many chromosomes. So some of the chromosomal diseases, Down syndrome, Turner syndrome, and Klinefelter syndrome, these are the some of the syndromes which affects in the gene. So non-distinction will take place. Last type is the of the gene mutation is the, these are the some of the types: point mutations, substitutions, insertion, deletions, and the frame shift. These are the types of the gene mutation. So this is the type of the gene mutations. These are the some of the types: point mutations, substitution, insertion, deletion, and the frame shift, same as this chromosomal mutations. So these are the about the mutation. So what is mutation? You have studied some of the definitions, history, characteristics, and the classification of the mutations. These are the classification of the brief classification of the mutation. So for more classes, subscribe to the Pavitra classes channel. Thank you.